Hello everyone, and welcome back to Plantastics Research. I've been recently doing a lot of different videos on how data is being collected and how it's being interpreted and how to use it in different formats. And I think this video specifically is going to be very important, especially because I'm going to be presenting my project to you guys very soon. Not in this specific video, but my long-term project that I'm doing for research. I hope everyone is doing well, and as I always say, let's get started. So specifically on today's video, we're going to be talking about recording data and experimentation, specifically how to perform an experiment, and what are some things to look out for, and what are some important key factors to look out for when you are performing an experiment. Experiments are extremely important, especially for science class, for everyday life. A lot of the society things that we use have been experimented with most of the time. Sometimes you may do an experiment in class, whether that's planting a seed or going outside and observing that many different forms of experimentation can be put out, and it's very important to keep track of your experimentation. And today I'm going to specifically show you guys how to do that. All right. So, as always, we have our standards. So we're going to be using SVO2, and also I've been noticing that, if you haven't noticed, we've been using a lot of botany standards, which are plants and things like that. And I specifically wanted to use botany because a lot of scientific aspects, especially environmental science, which I have been doing a lot of, especially things like plants and outside. So I wanted to continue to use the remnants of the standard from the botany, which is the study of plants. For our standard. So you'll see a lot of SVO specifically throughout the next few weeks. So our standard is to obtain, evaluate, and communicate information to delineate the plant divisions based on our current plant phylogenetic and taxonomic principles. And we won't be focusing specifically on this part, but I did want to specifically go to this: analyze and interpret data to develop models. Now, we aren't going to be developing models, but we are going to be developing something similar to a model. But it's a way to keep track of your data and understand the full aspect of how your data is being kept, especially if you're performing a long-term experiment. And I'll explain what a long-term experiment is in just a second. Key terms. As always, we have our key terms. So our first key term is spreadsheet, and that is a computer application for an organization of data. I'm going to be presenting to you guys a spreadsheet towards the end of the video, so please make sure you check that out and stay for that, because it's going to show you how certain data is kept in a real experiment that could be done and performed. Variables. Independent and dependent variables. These are two things that we're going to talk about a lot in this video. Actually, this is pretty much what most of the video is about and why these two things are important. And there's one more thing that you'll find as well. And of course, experiment. What is an experiment? An experiment is a scientific procedure taken to make a discovery and test a theory. And a lot of the times the theory is represented, or you've heard it represented, as a hypothesis. And that is an opinion or a thought that is given, or an idea, and you want to put that into play to test, is this true? Is this not true? So you definitely want to make sure you take into account of that, because it's a very important key factor. And discovery, of course, maybe you may have a general idea and you may not, and you may be unsure, and you would like to see the outcome or what are the results. So it's very good to look at the discovery aspect as well of an experiment. So our first variable, which is the independent variable, and this is a term that you're going to hear a lot, especially when you're doing experimentations. So the independent variable, what is it? So the independent variable is sort of like the starter point. This is So this is what causes the changes in an experiment. So this is what originally is put into. So we're gonna look over the example right here. So each of these bowls filled with soil, and I'm assuming plant seeds, was filled with a different type of liquid. So this was filled with tap water, bottled water, sugar, and this was filled with another substance. And all of these were tested to see the growth of which one would grow. The the independent variable would be represented as the amount of water. That is something that would be causing the change with the amount of water, or not even that, the type of water as well. The type of water, specifically in this specific visual, shows the amount, the type of water that is being used. So basically, you can ask the question, which one would make the plants grow faster? And that would be like your, which one would make the plants grow faster? And that would most likely either be the tap, the bottled water, in very slim cases the sugar, because sugar is something that helps the plants grow, and whatever this substance is. I'm sure this is probably maybe vinegar or milk. I forgot what it was, but yes. So it's basically what causes the change, and what do you add to therefore present a difference to the outcome. Now let's go to the other variable in the experiment, and one of the most important, the dependent variable. And this is the changes that occur after the independent variable has taken its course. So basically, if we look at this example right here and we read it, it says the different sizes of plants after they have had the substances added in the independent variable step. So each of these, as a result of the let's take into account the other picture with this picture. Let's say this was the bottle, the tap water, this was the bottle, this was the sugar, this was the vinegar, and this was milk made. Let's say that each of these would have a different, have a change. Some may not grow because, you know, milk wouldn't necessarily be a growth or provide growth for a plant, but maybe the tap water would grow because it's actually water, and plants need water along with sunlight and soil to therefore grow. So the dependent variable are the changes that are made in the experiment, and they are very important to keep track of, especially because the dependent variable are your set data. The size, the shape, sometimes, the shape sometimes, color, which is a chemical change, but we'll discuss that in a different video, and things like that. So you want to keep in mind everything that is going into account when you're explaining, or you're looking at and observing, which is also a very important word to keep in mind of when you're observing your changes that are being made. Constant. Now, the constant is one thing that we did not mention in our key terms, and the constant is extremely important to keep in mind. The constants are the things that are way before the dependent and independent variable. These are the things that start the experiment most of the time, and are the things that are kept the same. The temperature in the room, let's say even sometimes it could be the type of substance used, but it depends on the experiment. All constants can change depending on the type of experiment. So if you were testing maybe not the substance of the plants that you were using to defer the outcome, let's just say you were doing a general plant on two different types of plants, they will be given the same amount of water and the same type of water, and the water would then turn out to be the constant, not the independent variable. So it's very important to keep track of what your constants are, and are you making sure that you're putting the exact amount into this, depending on the type of experiment that you are presenting, because it's very important to note that your constant can and sometimes will differ from your dependent variable. So, as I have an example of a thermometer to represent the temperature, or is it a cooler temperature, is it a hotter temperature? That is determined, but that will always be the constant, because it's now you can, there are some experiments where they may be placed in different temperatures. That would then not become a constant, it would become a independent variable. So it's kind of, you want to make sure that you understand the difference between a constant and a dependent variable to make sure you don't get those mixed up. Now, we're going to talk about one of the, a good way, an example of keeping track of your data. Because data, sometimes you can have a small amount of data, sometimes you can have data, but not exactly a pattern of data, so it wouldn't always be high numbers. And you want to be as real as possible with the data that you present, and don't worry about how it may be presented, because all different data types may not be the 100% accurate, but in your case, in your standpoint, and if you're able to explain and back up what you've done for your data, every outcome could sometimes be just as different from the other. And it's very important to discuss that. And when I present my data findings and things like that to you guys, which I'm not doing right now, but I am going to show you the outline that I'm going to be using for it. It will then give you guys a general idea. So let's go to the link that I have here. Yes, so see here. Yes, so I don't know if you guys are able to see it. I deeply apologize, but basically what it has here and what I'm sharing with you guys is a, oops. Yes, okay, I apologize, guys. So yes, it is an experiment that I just thought of in my head. So basically, we have water, milk, soda, and then we have the days that we leave them out and how long, and it would be our type would be, our constant would be, let's say rose petal, a rose plant. So a rose plant would be our constant, or that would be the thing that is being used, the main thing that we're being used to present the experiment. And let's say if we added water to the soil, we added water to the soil. So let's say added soil, or added to soil. I think that would make a little bit more sense. I am so sorry if you guys aren't able to see it. I have the presentation opened very small, so hopefully you're able to see it. If not, that's okay. I can explain it. But so if we added water to the soil after day 1, it would still be 0 centimeters. Milk after day 1, it would be 0 centimeters. Soda after day 1. Now, I skipped to day 5, and I said that the plant that was watered would have 3 centimeters grown. The milk plant would still have 0 because, milk is not generally used to water plant. And the soda may have, let's say, I just made an estimation of maybe, let's say, less than 1 centimeter, just because, it may have a lot more, may have general water, but it probably wouldn't. And then I just made a loaded estimation here to say that the water would probably be 8 centimeters after 10 days, and the soda would just, imaginally be 4 centimeters, just to defer the data. But on a real standpoint, there are real things that you can add to different experiments to make them, to make the difference. So if we go back, yes, so that is basically what a spreadsheet is, and that is how my data is going to be laid out. It's going to be laid out a little bit more of a better format, but that's because that was just, data I kind of came up with, but it was to show you guys how the data is represented on the spreadsheet. And a few websites that you guys can use are Google Sheets and Excel if you have that software downloaded on your computer. And as I always say, thank you guys for coming to view the information with me. I was glad I was able to share this. This is probably one of my favorite videos because I was able to explain, the different types and aspects of how data and experimentation is not only collected, but how it's represented and interpreted. So I hope everyone has a wonderful day, and thank you for viewing Plantastic Research. Goodbye.